Thank you very much. That is my introduction. I'll go right to it. So today we are listening to all the tumor and phylogeny. So I was also working in the same line. So for genetic disease, you want to look at both mutation and expression. So expression, you look at RNA. For mutation, you look at DNA. But DNA at this point is we are working at a sample or population level. So we know this patient has this mutation, but we haven't been able to map which cell express these SMVs. We could do single cell exome sequencing, but so far the coverage and the depth has not been sufficient to get enough information. So why not just use RNA? We are doing single cell RNA sequencing, and there are algorithms that can get SMV information from these, so like deep variant or recently schematic. Also, now we have long-read sequencing, which captures the entire RNA sequences instead of fragments. So how well can we do it? So this is what I wanted to test. So we made an algorithm called scVarID. Taking a BAM file and a VCF file, instead of count metrics, we generate mutation count matrices. And we can do this as long as there's a BAM file and a VCF file. We can use this to, what I did is to sort what I call a useful SMV, which are present in sufficient proportional population with sufficient depth. Then I'm using the allele fraction to see which is the fraction of altered allele counts over the entirety of the counter. And we visualize it and see what kind of information we can get. So in order to do that, I need a gold standard VCF as well as a single cell long read sequencing. Fortunately, I found one from Genome Innovato individual HG002, which contained 4 million SMVs. When I tried to map this on a single cell long read sequencing, I mapped about half of them, so from 4 million to 1.7 million. When I tried to get a useful SMV, which I defined as I can verify it to exist in a 5% of the population with the depth of 10x or more, I got 459, which is represented in the heat map here. So this is about 10,000 cells with 459 SMVs with the allele fraction from 0 to 1. What is clearly evident is the giant red block on the top, which represents the allele fraction 1, which will most likely represent the genetic, the germline variants, which are probably mutated in both copies, which can only express the altered variants. What you can also see is these horizontal lines and blocks, and we will explore this further. So after removing some obvious homogeneous SMV distribution, which are, for me, not so much interesting, I was left with the 320 variants. And when I looked at it closely, these map to only 5 genes. So 251 variants belong to 5 genes, and they're all in HLA1 class. So HLA genes are known to hypermutate, so these seemed likely. And the heat map I'm showing you here is the gene of HLA-DQ1B, which I have identified 72 SMVs from the gold standard from about 3,000 cells. From this pattern, there is so much information that I need to go slowly. So to the left, we have SMV, but instead of clustering, I ordered it by the chromosome positions. To the column, then I have cells which I have clusters. What you see is, and I have clustered these SMV patterns based on the allele fraction values, and it clustered into 2 nicely. What you see is an alternating pattern of SMV presenting as allele fraction 1, which means they're all present, or 0, meaning SMV is not there. Only in the middle, the cluster of cells express the allele fraction of 0.5, meaning that in a natural state, there is an allele-specific expression. So what I meant is, in the top is the proposed or possible presence of SMVs across 2 arms. So from the left column, then it is only expressing 1 arm, which shows that these SMVs are present in the allele fraction 1. On all the SMVs in the other arm are not expressed, which means the allele fraction of 0. The exact opposites are happening on the other columns. Only the second arm is presenting, so that's why the SMVs presenting the second arm has allele fraction 1. And in the middle, the both arms are presenting. That's why the allele fraction of 0.5 is happening. So this is a reference individual. So we also had a short read sequencing. So I was wondering, instead of long read, we have so many short reads. Maybe we could do this on the short reads as well. So we apply, same idea, BAM file, FreeCF, we mapped it. We are looking at HLA-A. So from the long read, we identified 44 SMVs from about 9,000 cells. From short read, only 6 SMVs from less than 2,000 cells. So the quality is definitely going down, but we got the same SMVs. What is interesting is that the SMVs that we got from the short read are actually only the top portion of the long reads. So it's only, as we suspected from the short read data, they are fragmenting and only the proportion of the genes are expressed, that are captured, then we could see very clearly that these SMVs from the short reads are actually the top portion of the long reads. But when we filter it to match it, we can cluster it all the same. So one specific arm has these 4 SMVs, the other arm has 2 SMVs, and there are allele-specific expressions to distinctly show that the allele fraction is near 1 or 0, depending on the state of the cell. So that's it. So we made this scVarID. We are generating mutational count matrices. And using these, we can do pretty cool things like allele-specific expression analysis or the haplotype determination. So this is a reference individual, so it's only one particular condition. We hope to apply this on a different conditions for a normal to tumor samples to see how these differ, or maybe there's an early detection of mutations in the normal cells that maybe something's funny is going on, tumor heterogeneities, or we could apply this on a different cell types and see if there are specific mutations prone to epithelial cells or the T cells and all the other things. So these are the references I've used. I thank for my funding. Thanks for your attention.
I was really following you, but I missed the part when you put the genomic coordinates to separate the contribution of the 2 alleles.
This one.
And you, yes, exactly. Just can you kind of try to say again?
Yes. So these are the allele fraction heat map. So I got all the reference counts and now the altered variant counts for individual cells. Then I just simply represent it as a ratio. So for individual cells, if it has an SMV, I assume the somatic variance usually infers to only the one copy is influenced and the other copy is normal. If everything goes okay, then both copies are transcribed. And if I'm looking at RNA, then I should get about 0.5 of allele fraction. But I get 1 or 0. Then, yeah. Yes.
So apparently all your genes are HLA genes, right?
Mostly, yes.
So, so could this be that this is specific to these types of genes, or do you think the same pattern will also occur in other genes?
I think pattern happens in other genes, but it's simply that I cannot see it, because in order to do this type of analysis, I would have more than one SMVs in a particular gene, but only on these 5 genes, there were so many SMVs that I could easily visualize and demonstrate. Probably a better sequencing technologies on depth. I can see even more information. That's probably on the data side. That's the limitation of the things that I can't show.
Are there any other questions? Let's thank our speaker again.
Thank you very much.